Hello, everyone. My name is Amy Klippinger, and on behalf of the PETA International Science Consortium and the Institute for In Vitro Sciences, I welcome you to today's webinar on in vitro assays for dermal safety and efficacy testing for regulatory and non-regulatory purposes. Today's speaker is Dr. Gertrude Amelia Costin. Dr. Costin works as a study director and manager of scientific services at the Institute for In Vitro Sciences. Her main area of expertise is in the use of in vitro test methods for the dermal safety assessment of ingredients and formulations manufactured by the personal care, pharmaceutical, and chemical industries. Amelia?
Thank you for the introduction. Hello, everyone. I would be happy to present today on a two-part presentation. The first one will address validated in vitro assays used for dermal safety assessment, and the second part will discuss assays that are not validated for regulatory purposes. I would like to set up the stage and talk about the integrated approaches to testing and assessment, or so-called the IATAs. They are an integral part of today's toxicology field, and particularly for in vitro toxicology that we are interested in. The IATAs take into consideration three parts, where part one is related to existing information on chemicals, part two is using weight of evidence analysis, and part three considers additional testing for the substances of interest where needed. When creating the toxicological profile of a chemical, a raw ingredient, or a formulation, all the data that are existing at any given time are taken into consideration. And they could represent human data, in vivo or in vitro data, physical chemical properties, and so on and so forth. Once all these data are analyzed, and a conclusion can be made, the products can be labeled and registered with the regulatory agencies. If that is not possible, scientists address the weight of evidence, or part two of the IATAs, and they perform further analysis to support the safety assessment of products. And finally, when needed, additional testing is to be performed, and today, in the field of toxicology, huge emphasis is put on alternative testing to animal testing, such as in silico methods, in chemical, in vitro, ex vivo, etc. So we will concentrate in today's presentation on in vitro methodologies that can be used for the safety assessment, particularly for skin. I would like to also make a point that as scientists working for industry, academia, regulatory agencies, animal welfare, or simply as representative of the general public, we are all in this process moving away from the animal testing together. We share the same perspectives, challenges, we share common goals, and we are trying to come up with good ways to work together to address the same goals. We can look at our work, collaborative work, from the perspective of either industry, testing labs, from the regulatory agency perspective, or from the consumer perspective. If we start with the industry or the manufacturer who comes up with a new idea of a product that doesn't have a label, doesn't have a toxicological profile, the goal is to have the product tested in using methodologies that are available nowadays. After the data are generated, analyzed, and the entire report on the safety profile is put together, they are submitted to a regulatory agency. Could it be EPA, could be FDA or OECD around the world, which makes a further assessment of the product with the final goal to avoid any incidents like this one where accidental exposure to a chemical can induce skin irritation or even worse, skin corrosion. Finally, after the product is deemed to be safe, it is put on the market, a proper label is provided to the product, and it can be used by a consumer. So in my presentation, in the first part, I would like to discuss about the impact of test substances on human skin. I will briefly review the animal test system that is used for acute dermal corrosion and irritation assessment. And I will detail on in vitro assays that are validated for regulatory purposes to address these two endpoints, skin corrosion and irritation. Finally, I will provide an overview of the testing strategies that use in vitro assays for skin corrosion and irritation endpoints. I know that it is of interest to this audience to talk about any limitations of in vitro assays, any typical or modified protocols that we can use to address safety assessment, any prediction models, and I will also provide a couple of examples of case studies. Let's talk about the impact of test substances on human skin. Human skin is the largest organ of our body, and it acts as a physical barrier against mechanical, chemical, and microbial factors. It is also an immune network, and it's our defense system against UV radiation to which we are exposed on a daily basis. Skin has a very complex structure, as you can see by the many layers that come into contact with substances. Stratum corneum, which is the exterior layer of the skin, is the one that limits our exposure to toxicants. However, when a skin corrosive or irritant material comes in contact with our skin accidentally, it can induce two different types of events. One is irritation, which is a reversible damage to the skin following exposure to a test substance. Irritation is characterized by erythema, the redness appearance, and edema, and it's usually attacking keratinocytes and dermal cells in the skin, which further lead to inflammation. When a corrosion material is coming in contact with the skin, this is an irreversible damage, which is visible by necrosis through the epidermis into the dermis, and is usually typified by ulcers and bleeding. Companies are doing their due diligence to address any corrosion or irritation potential that their products may induce to humans. The testing is necessary to put the correct label on the product in the end and instruct the users as of the risks of using the product and precautions that they should take should any exposure happen. So testing is critical for the registration and labeling of chemicals, for their transportation, also for occupational safety. Workers handling large amounts of products in bulk in the manufacturing setting are more exposed to the possibility to get in touch with large amounts of products, and they need to be instructed properly how to take care of themselves should that happen. Finally, proper labels should be provided to cosmetics and household products. In brief, I would like to go through the DRACE test, which is the animal test system used for acute dermal corrosion and irritation assessment. It has been known since the 40s, and it's a method that uses albino rabbits. The assay endpoints are erythema and edema, and the assay control is the untreated skin of the test animal. The test method is used for the evaluation of corrosion and irritation potential of test substances. There are, however, limitations. Like with any test system, we have to take those into account for a proper assessment. The rabbit and human skin have different physiological properties and responses to environmental and chemical agents. As such, some compounds are more toxic to rabbits than to humans, and vice versa. The DRACE rabbit skin irritation test has been criticized over time for its over-predictions of human skin, and has also faced a debated ethical issue of the in vivo test due to the animal suffering and discomfort, especially with corrosive materials. The test system and the method is covered by OECD Test Guideline 404, which has been updated last time in 2015. In brief, this is how the assay is performed. 1 to 3 animals are tested in the test in a sequential testing. Should any events happen earlier in the 1 animal study, no other testing is continued, not to subject the animals to further suffering. The test substances can be tested either as liquids or as solids. The exposure times are relatively short for the skin corrosion, 3 minutes, 1 hour, and 4 hours, and longer, up to 4 hours for skin irritation. These time points are very important to keep in mind for the rest of the presentation because they were used for the validation of in vitro methods to address the trace test for skin irritation and corrosion. The animal is followed for an observation period up to 14 or 28 days, and the scores for erythema and edema, as outlined in these 2 tables, are calculated for each animal, and they are used to generate a score, an irritation score, that is further used by regulatory agencies to generate labels for the product. Due to many reasons that I outlined already, industry, academia, regulatory agencies, animal welfare, trade agencies came together and thought about other ways to address the safety or dermal safety and to move away from the animal test. There are a couple of methods that are validated for regulatory purposes for the 2 endpoints. For corrosion, 3 methods are available currently: the transcutaneous electrical resistance test, which I will not discuss in this presentation, membrane barrier test method, Corrosion, with OECD guideline 435, the reconstructed human epidermis test method with OECD number 431. For skin irritation, we can also perform the reconstructed human epidermis test using reconstructed tissue models, and we have the OECD guideline 439. I will start with the Corrosion assay, which is the simplest of the methods I will discuss in this part of the presentation. It is an in-chemical assay that uses an artificial membrane designed to respond to corrosive substances in a manner similar to the animal skin. The assay endpoint is the time in minutes that is required for a test substance to penetrate through the Corrosion biobarrier membrane and to produce a color change in the chemical detection system. As with any assays, negative and positive controls specific to the assay need to be included with every single study that is performed. The assay is applicable to assigned UN packing groups to corrosive materials or to verify if a test material is not corrosive. One of the limitations of the assay resides with the pH of the test substance to be investigated. Usually, if the pH is between 4.5 and 8.5, those materials may not qualify for the assay because they do not induce a color in the color detection system. The OECD guideline 435, which covers the Corrosion method, has been updated in 2015. I would like to address a couple of technical notes that are very important when the assay is performed in the lab. One of the first questions that we ask is if the test substance qualifies for the assay. Usually the test substance is added to a tube containing this yellow color chemical detection system. If the product induces a change in the color of the chemical detection system, we can capture it in this modification, and therefore the product can be used in the assay. If the test substances do not induce this color change, they cannot be tested using Corrosion, and some other alternative methods should be considered. Next, the test substances are added to 2 test tubes to determine the appropriate packing group assignment. Using a chart from the manufacturer of the kit, In Vitro International, we assign either a category 1 to the test substance, which will be evaluated up to 4 hours, or a category 2 test substance, which will be evaluated for up to 1 hour. Another consideration is the setup of the assay. Usually, the assay is performed in a hood with a strong and white light, and we use styrofoam environment just to be able to detect the formation of the color. Although faint in this picture, you can see a red color in these tubes, which means the test substance penetrated through the membrane into the color detection system, and it already induced a modification. In general, the assay can be performed within a day. We receive all the ingredients of the kit together from the manufacturer. We prepare the biobarrier in the lab, and the biobarriers are fixed on the top of a vial that contains the chemical detection system. Then each test substance is added to 4 replicate biobarrier membranes, and the chemical detection system vial is continuously monitored for the first 10 minutes. And we record the time in minutes that it takes for a test substance to penetrate through the chemical detection system and induce a color change. Based on that time, and knowing if the materials were assigned for a category 1 or a category 2 type of assay, packing groups from 1, 2, and 3, or not applicable, will be assigned to products. This type of assay can be used for transportation purposes, and it is accepted by the Department of Transportation for that purpose. The next assay that in the next slide, I would like to discuss the OECD test guideline 431, which is the reconstructed human epidermis corrosion assay. The assay has been validated for the purpose of testing liquid materials, semi-solid, and solid materials, and it is assumed to be applicable to mixtures also. In this particular table, I would like to emphasize the performance of the skin models that are validated for this particular type of assay. And to bring to your attention that at the beginning, when the assay was validated, the models epidermis, kinetic, and EPCS it did not perform very well for subclassification in comparison to the EpiSkin model. However, this caveat has been overcome recently, and I will explain how the prediction model has been modified. In this slide, I would like to share a set of data for the Corrosion assay in comparison to in vivo data. The results have been generated in collaboration with the company Johnson Diversey at the time. The company changed the name recently, and it's known to be Sealed Air these days. The data have been reported at an SOT meeting, so they are not confidential at this point, and they are available in the public domain. So we tested in this study 7 products that had animal data, and they were corrosive, they had extreme pHs, and they were in the high pH range. For these products that are highlighted in yellow, the 2 test systems, the in vitro corrosive test and the in vivo animal test, predicted the same category of corrosive or non-corrosive. The products that are pointed to with red arrows were the products for which the in vitro test system predicted non-corrosive products as being corrosive. So we are talking about a potential over-prediction by the in vitro test system as a limitation. We approached the company and asked why only these products have been impacted and why only for those the in vitro methods over-predicted. And they were able to isolate the fact that the products had high level of solvent, more than 15%, and resulted in a more conservative classification when using the in vitro assay. It is very important for industry to share this type of work such that regulatory agencies, other companies, public in general is aware about some chemistries that may not be addressed by the in vitro methodologies. Moving into the in vitro reconstructed human epidermis test models, I would like to emphasize that there are several tissue models that have been validated for regulatory purposes. One is the epiderm tissue model from the U.S. manufacturer PETA Corporation, SkinEthic and EpiSkin from the manufacturer in France, and also LabSide and EpiCS with other manufacturers around the world, in Japan and Germany. These tissue models are reconstructed from human keratinocytes, and they display a number of layers similar to the architecture of the human skin. Usually, the products are sent to the end user laboratories around the world with a quality control document from the manufacturer stating that the products have passed the manufacturing criteria, and that they have the capability of limiting the exposure to a test substance having the stratum corneum fully developed. That said, when used in the assays, the tissue models have to be subjected to the proper negative and positive controls. The first assay that I would like to discuss is the skin corrosion assay used in reconstructed tissue models. As I explained, 4 models have been validated for regulatory purposes. The assay endpoint is the tissue viability, which is assessed by the reduction of the vital dye MTT, which is a yellow color material that is reduced by viable cells and is transformed into a dark purple dye in the end, which is a measure of the viability of the tissues or all the cells. The assay controls are different based on the tissue model that one can use. So it can be, for example, for the negative control, either water or sodium chloride, and for positive control, it could be potassium hydroxide or acetic acid. On the applicability side, the results of the assays can be used for regulatory purposes to distinguish corrosive materials from non-corrosive. Furthermore, the test method allows for the subcategorization into 1A corrosive materials and a group of 1B and 1C. One of the limitations of the assay is the fact that the method does not allow to discriminate between skin corrosive subcategories 1B and 1C according to the globally harmonized system, GHS, due to a limited set of well-known in vivo corrosive category 1C chemicals. The OECD guideline has been updated lastly in 2016. As with Corrostetex, I would like to address a couple of technical notes that are critical for the good performance of the assay. The first question that we ask when working with substances is, do the test substances reduce the MTT directly? In certain situations where the test substances manage to reduce on their own the MTT from a yellow color to a purple color, we have to address that properly in the lab because it is an artifact of the method. For example, the negative control is not expected to reduce MTT. However, as you can see, this test substance on its own, in the absence of the tissues, reduced the MTT from a yellow color to a purple. In this situation, we use in the lab what we call kill controls. The kill controls are the tissues that were subjected to freeze and thaw cycles, 2 or 3, basically with the goal to kill the tissue. That means that that tissue does not have the capacity, metabolic capacity, to reduce the MTT on its own. Therefore, any reduction of the MTT on a kill control tissue, it is assumed to be due to the test material that will reduce the MTT on itself. This is very important to keep in mind when we are working with materials that cannot be rinsed from the surface of the tissue. So in this situation, it is critical to consider kill controls in the experiment and to address the calculations for the data properly. Other assay considerations, anyone can work with different types of products. For the liquids, we use positive displacement pipettes. For viscous materials, we can use dosing devices applied on the top of the test material onto the tissue. The purpose of the dosing devices is to ensure that the entire surface of the tissue is exposed to the test substance of interest. Finally, per OECD guideline, the solids are applied to the tissues using a calibrated spoon for a 25 milligram dose. This is the typical protocol that can be used for the skin corrosion assay. For the purpose of this presentation, the pictures display one of the tissue models, specifically the epiderm tissue model from PETA Corporation. As I mentioned before, there are 3 other models that one can use to address skin corrosion. Usually the tissues are received in an agar plate for transportation purposes, and they are placed into the culture for about an hour to acclimate them from the shipping to the incubator condition. After 1 hour, the media underneath the tissue is refreshed with warm media, and duplicate tissues are treated with the control and test substances for the exposure times that are specified for each tissue model in the guideline. For this particular tissue model, 3 minutes and 1 hour are used. After the exposure is completed, the rinsing procedure is followed to remove the control substances and the test substances from the surface of the tissues. Then the tissues are incubated in MTT for 3 hours to allow the reduction of the MTT by viable cells or not by the cells that were impacted by the toxicity of the product, such in this case. MTT is then extracted into isopropanol for 2 hours, and a volume of the extracted MTT in isopropanol is plated then on a 96 well plate, and the optical density of the samples is captured using a spectrophotometer. Those optical density values are then converted into viability values of the tissues. Depending on the tissue model, a different prediction model may be considered, where in general, we can separate corrosive materials from non-corrosive, and we can subcategorize category 1 corrosives from 1B and 1C, as I explained before. As I mentioned, EpiSkin model was the one that was capable to subcategorize initially during the first round of the validation. The tissue models epidermis, kinetic, and EPCS have been brought to the same level of prediction for subcategories a couple of years back in 2015, when manufacturers got together and looked retrospectively at the data. Based on the manuscript that was published in 2015, the OECD guideline have included and considered these cutoffs to separate category 1 materials from 1B and 1C. The cutoff values for epidermis, kinetic, and EPCS were 50% and 15%, whereby the combination of those corrosive and non-corrosive materials were separated. Using a 25% cutoff viability value for epiderm model, 18 for kinetic, and 15 for EPCS, we can now subcategorize using these tissue models into 1A and 1B and 1C. I would like to give an example from our work, again, with Johnson Diversey in 2011, using this time acids. So we tested several products that are highlighted in yellow in this table, and we compared in vivo data with in vitro data generated using the epiderm tissue model. So in certain cases that are marked in green, the 2 assays actually agreed on the same corrosive or non-corrosive classification. There were some cases pointed here with the red arrows in which the discordant classifications have been obtained, where again the in vitro model over-predicted non-corrosive materials. Again, performing an analysis of the identity of those over-classified materials, the company shared that in general, for an acid reserve that is more than 1.78, the agreement between the in vivo and in vitro method was only 25%. So we isolated chemistries that may be over-predicted in vitro compared to the animal data. In general, the in vitro assays perform very well for ingredients. Mixtures are more complex in their impact on the skin, and we need to be aware of chemistries that are more challenging to address in vitro. I hope you remember the example of comparison that we had for the Corrosion assay a couple of slides back, when we used bases with extreme pHs and compared the Corrosion assay results to the animal test. We did the same exercise here for 12 products, for which we compared the in vivo trace data to the epiderm-based corrosion assay. So for the most part, for the parts in yellow, the two assays agreed on the same classification. 8 out of 12 products were in agreement. However, there were 4 of them for which the in vitro system over-predicted compared to the animal test system. To our surprise, those formulations were the same as for Corrosion, the ones that had high level of solvents, more than 15%, that resulted in a more conservative classification. So the in vitro assays agreed between themselves, and they overclassified, providing the manufacturer a more conservative way to label. From the regulatory perspective, it is very important not to miss any potential corrosive materials with the risk of sometimes over-predicting or over-labeling. From the consumer perspective, that this is the conservative way to go. I would like to share another example from a poster presented last year at the Society of Toxicology meeting, where by the company SCJ, where the Corrosion assay and the epiderm-based corrosion assay have been compared against the in vivo classification of products, or in the view of the globally harmonized system additivity formula. This was a very interesting set of data that concluded that the Corrosion method appeared to be a better assay to clearly distinguish among all packing groups without overclassification. This is critical for the transportation of those products. In terms of the epiderm skin corrosion assay, it was concluded that it did not correlate with the globally harmonized system classification for extreme pH driving corrosivity, and was unable to distinguish between UN packing groups 1B and 1C. Interestingly enough, speaking of isolating different chemistries that are more challenging to address in vitro, the work presented concluded that sodium hypochlorite containing mixtures may be under-predicted based on the effect of color change in the Corrosion assay. We consider this sharing of data and knowledge extremely critical and important in the industry and with regulatory agencies, so we are all aware of what challenges we face and find solutions together. Thus far, I provided examples only of over predictions of products by the in vitro test systems. Here's an example of under-predicting. This is work performed a couple of years back in 2015 by AkzoNobel using phthiamines. So the company performed the corrosion assay and the skin irritation assay, and they compared the in vivo data to in vitro data. In only one case, the two assays, the in vitro and in vivo platform, agreed on a product. In other instances, the in vitro model under-predicted corrosive materials, whereas the animal test predicted a corrosive outcome. In looking into reasons as of why this may be the case, it was determined that fatty amines are recognized for their severe irritating and corrosive effects based on an inflammatory event that happens. It's a delayed severe inflammatory event that happened in the animal. However, this is not captured in vitro because the assays are very, very short. Remember that the corrosion assay is 1 hour exposure maximum, or 4, depending on the tissue model. So we as a community, I think, have a duty to look into how various particular classes of products can be addressed in vitro, knowing that some may be more challenging in being under or over predicted. So in this situation, the question is, can the data be considered for a submission for a regulatory agency? And if not, what modifications can be performed to the assay, which is regulatory validated, to make it work, to make it capable to address, in this situation, fatty amines? I would like to provide case studies for the in vitro assays. And I would like to provide it from the perspective of the study director and share with you what we look for in a data or a report. We look to see if the assay was a valid assay, if it met the criteria specified in the protocol. We also question, are any results equivocal? And if so, are they the result of a nature of the test substance, of a technical error, of tissue performance? Is a repeat trial necessary? Is a follow-up assay necessary? How is the test substance labeled based on the data generated? Can the data be used for labeling a particular class of substances with confidence, or are the assays not properly addressing the endpoint of interest? Since we talked about corrosion assays, let's start with the first example and discuss implication of tissue loss during the assay. Let's take the example of substance A. What we know about it is that it was a clear colorless liquid, and in the workbook notes, we noted that the second tissue treated with the test substance for 60 minutes became dislodged from the cell culture insert and may have been lost. In looking at the data, what we noticed for the test substance A for the 60-minute exposure is that the optical density value of the tissue that we noted was possibly lost is very low compared to the optical density value of the first tissue. Overall, the prediction for this assay, based on the 3-minute result, is that it's a non-corrosive. However, the 60-minute calculation put the test substance in a corrosive category. Still, we have this problem and issue and concern with the tissue that may have been lost. So what do we do next? Option 1 would be to be conservative and label the test substance as skin corrosive, taking into account both values for the 60-minute exposure. In that situation, the outcome would be that the test substance is corrosive. Option 2 would be to exclude the second value, and since we presume that the test material, the tissue was lost, and recalculate. Here's the risk if we remove this value. The test substance would be predicted non-corrosive by both time points. However, we need to look now into this value that we generated in this case for the viability of the tissue, which is 15.2%, which is interestingly very close to the value that we generated for the positive control of the assay, which is deemed to be corrosive. Keeping that in mind, option 3, and probably the best in this situation, is to repeat the assay. The next test method that I would like to discuss is the skin irritation test based on reconstructed human epidermal models. The OECD guideline for this test method is 439, and it was updated 3 years ago. As with the corrosion assay, several tissue models have been validated for this purpose. The assay endpoint is the same, the MTT, addressing tissue viability. Specific assay negative and positive controls, as I said, are included with every study. On the applicability, the results can be used for regulatory purposes to determine the skin irritancy of test substances, either as a standalone replacement for in vivo skin irritation testing or as partial replacement within a tier testing strategy. One of the limitations of this assay is that it does not allow the classification of test substances to the optional UN GHS category 3 for mild irritants. As with the other assays that I presented, I would like to address pre-assay considerations and technical notes. One of the key questions in this assay is to ask if the test substance interacts with the mesh in the case of liquid test materials. The volume that is used in this assay is very small, 30 microliter, compared to the corrosion assay, which uses 50 microliters. So to spread the test material on the entire surface of the tissue, it is necessary to apply onto the test substance a nylon mesh. There are situations where the nylon mesh can interact with the test substance and affect the tissues just by that event alone. So we need to test for this particular interaction between the test substance and the nylon mesh before we even start the assay using tissues. In this situation, the mesh can be used, whereas in this situation, it cannot be used because the nylon mesh has been dissolved by the test material. The rinsing procedure in this assay is very specific. It is outlined in the OECD guideline. It involves the removal of a mesh after the 5th rinse, for example. The tissues have to be observed for any residual test materials using a scope. And careful handling is exercised when we have to remove test substances from the tissues using sterile Q-tips. Also, in the observations under the microscope, we make notes in the study workbooks of any unusual occurrences on the tissues. Like in this case, the tissue would show what we call blisters, which could be a result of the toxicity of the product. This is the typical protocol used for the skin irritation test performed in vitro. The main steps as used for the corrosion assays are followed. I would like to emphasize the key differences between the assays. In this method, the tissues are pre-incubated before being treated with the test substance. They are incubated in the media first for 1 hour, and then with media change for overnight to acclimate the tissues for a longer period of time that they will spend in the incubator. Triplicate tissues are used for the assay. A rinsing procedure is very specific, and after the rinsing is completed, the tissues are incubated in the media for 24 hours. After 24 hours, the media is changed, and the tissues continue their incubation in fresh media for an additional 18 hours. So the assay uses a short exposure of one hour and a long post-exposure of 42 hours. The post-exposure is necessary to address any delayed irritation events. We know that irritation takes a longer time to show in the tissues compared to corrosive materials, which penetrate rather fast and induce very significant damage to the tissues in a short time. After the post-exposure is completed, all the steps with MTT and reduction, extraction and reading of the optical density values take place. Again, the optical density values are converted into viability values, and the cutoff used for this particular assay is a 50% viability. As such, if the viability of the tissues is less than 50%, the product is predicted to be an irritant to the skin, and the UNGHS category could be either 1 or 2. If the corrosion assay has not been performed, we do not know if this product would be either category 1 or corrosive or category 2 or irritant. I will come back to this concept a little later. Finally, if the mean viability of the tissues is more than 50%, the products are deemed to be non-irritants, and they are considered a no category for the purposes of UNGHS categorization system. I included in this slide one of the available videos that explain this particular test method in detail, should you want to learn more about it. I would like to give an example from the pesticides world. This is work performed by BASF, particularly on agrochemical formulations. And I pointed in red situations where the skin irritation test under-predicted in this case the in vivo classifications, and in black where they agreed on the same category. You can see for the skin corrosion test, there was high specificity and accuracy for the in vitro test, whereas for the skin irritation, these values are a little lower in comparison. I would like to give you examples of case studies. And this one asks the question, is this a valid assay? Thinking of the skin irritation test, I would like to provide to you the criteria that consider an assay valid. We know that the mean optical density value of the negative control should be between 0.8 and 2.8. The standard deviation for the viability value should be less than 18%. And the viability of the positive control should be less than 20%. So let's look at this set of data. These are the numbers for the negative control. And we already see that we have two values here that are above this cutoff value of 2.8. Actually, only this one is higher. So what we question in this situation is this assay valid for from the positive control and negative control perspective. So outlined here are the data for the positive control. These are the optical density values, and these are the viability values. And we already detect two issues where the viability values of these two tissues are above this 20% cutoff value. So while we ask, do we have a valid negative control in this case? No, because one of the values of the tissues were above the cutoff that is acceptable for the assay. Is this a valid positive control? Clearly not. So what happens next? Next, we would likely repeat the study and analyze the data again. Case study number 2 addresses variable results. What do we know about test substance A? It was an opaque green liquid, viscous liquid, and the workbook stated that there were residues of the test material on the tissues after rinsing. So these are the data that we obtained for this particular test system, test substance in the 3 tissues. As you can see, the values are very different: 48.3, 9.8, and 64.7% viability. We also have a high standard deviation for this product. So with the mean viability of 40.9, this particular substance is considered to be a skin irritant. And the standard deviation that we obtained is indicative of variability, since we had residues of the materials left on the tissues. So it is likely that the varying amount of the residues on the tissues caused varying cytotoxicity responses. In the assay, none of the other treatment or control groups showed variability, high variability. So the cause of the variability for this test substance A is likely related to the nature of the test substance and possibly cause for overprediction. So in this situation, we're facing an equivocal response. What do we do next? A study director faced with this dilemma will inform the sponsor and will ask the proper questions. Does the assay mimic a human exposure with exaggerated persistence on the skin? In other words, the residues are to be expected when a person is accidentally exposed to this product. A conservative approach would be to label the product as an irritant and relate to other data that may be available for this product that may indicate skin irritation. Since the test material is predicted to be a skin irritant, the next question to be asked is the product potentially skin corrosive? So the skin corrosion assay should be performed or considered. Finally, another conservative approach would be to repeat the study and reevaluate the results and look for any potential variability to be reproducible, which would guarantee a discussion with the sponsor again. Since we talked about skin irritation and corrosion together to be addressed using in vitro assays, I would like to explain a self-correcting integrated testing strategy that can be used to address these two endpoints. If data are available on a test substance that would indicate a potential for corrosion, such as pH or other physical chemical properties or other data that have been generated before, the test substance can be tested in vitro using the in vitro corrosion assays. If the test substance is predicted to be corrosive, it can be labeled like that. This is the top-down strategy. If the test substance is predicted to be non-corrosive, we do not know if it will be an irritant to the skin. So the bottom-up strategy can be used where the skin irritation test is performed in vitro. If the test substance is predicted to be an irritant, it will be verified if it is also a corrosive. So the top-down strategy will follow. If the test substance is predicted to be non-irritant, it can be labeled like that. So this is a self-correcting strategy that ensures that the right labels are put on the product and that these events do not take place when a user is exposed accidentally to this product. Finally, I would like to emphasize that companies use these strategies in different ways, knowing their chemistries. Again, public domain information, the strategy is used by pharmaceutical companies where one of the companies always starts from the bottom-up strategy and follows with a top-down if needed, and another one starts always with a top-down strategy. So it all depends on the type of product and what knowledge the company has before starting testing. In the grand scheme of the integrated approaches to testing and assessment, or IATAs, all the information, all the methodologies that I discussed thus far will be integrated in part 3, where this additional testing using in vitro methodologies will take place should all the other work not be supportive enough to guarantee a proper labeling. Getting back to the first slide of my presentation, I hope I convinced you that multiple parties share the same perspectives, challenges, common goals, and clearly work together. From the industry and manufacturer perspective, trying to put on the market safe products and efficacious products, the manufacturer is not alone. They work with academic labs, with animal welfare groups and trade associations to ensure that no critical adverse effects occur to the skin of a user. Safety testing labs have a high order of their mission to put on the market available methods that are high throughput, sensitive, reproducible, specific, reliable. This is a very ambitious goal. And by this hard work in collaboration with all the others, it was possible to validate in vitro methods. Finally, from the regulatory agency perspective, and in this case, my example comes from the US EPA, it is critical to have the support of agencies to move away from the animal testing. And I reference here one of the programs that address the eye irritation endpoints for the labeling of pesticides in the United States. The letter from the head of EPA, Jack Hausinger, a couple of years back, supporting the non-animal testing strategies. And finally, recently, the TSCA work that is taking place in the United States to incorporate alternative methods into the labeling process. So if we get back to the DRAISE scale that addresses products from anywhere from corrosive materials to non-irritant materials, we know that the animal is capable to address all these, this entire range. However, the products that can be tested cover themselves this range from corrosive to non-irritants, and it is rather challenging to address an up to 28-day assay using in vitro methods that go possibly only up to 24 hours of exposure. However, that is possible, keeping in mind that possibly multiple in vitro assays should be used in a tiered testing strategy or integrated testing strategy to address this, this wide range of dermal irritation or corrosive potential. So from what we discussed thus far, we know that this end of the spectrum of corrosive products can be addressed using in vitro skin corrosion assay. For the severe irritants or non-irritant materials, we can use the in vitro skin irritation test. However, there is this middle ground, as also shown in this MTT response, where the results may not be so definite, like yes or no, as addressed by validated in vitro assays for regulatory purposes, where the products could be either moderate or mild in their irritation potential to skin. So question is, what assays can be used to address this middle range? And the response to this question will be addressed in the second part of my presentation that will follow shortly. Thank you for your attention.